slide with a few problems, a few fascinating, deep, central, I believe, problems in evolution. These are problems that I have thought about, okay, a little. And I'm going to tell you. So this is the part of the talk where I stick my head out, okay, you know, and you take appropriate action. So, but I want to actually, you know, just to show that I've been watching Nagylaki's talk. You know, I just want to remember something from him. So my full name is Christos H. Papadimitriou. I don't know if anybody knows what H stands for. It stands for humility. Okay, right? So, actually, hard is too. So that's, the role of sex is a well-known, interesting problem. So there is 15 years ago, Nick Barton and Charles Wolfe wrote a famous survey, a famous paper, basically setting it up, and what I want to say, I mean, I want to connect this with what the way I ended the first half of my talk, with the two genres of heuristics, evolution-inspired heuristics, simulate annealing and genetic algorithms. So what I'm saying is that the ubiquity of sex, the success, the empirical fact of sex's success in life, makes the apparent advantage of simulate annealing over genetic algorithms harder to explain. Here is when sort of, what really got me into studying evolution is meeting Adil Livnat, but that's about 7 years ago now. And during our first discussion, which lasted an inordinate amount of hours, I explained to him my frustration with not understanding this fact, okay, that simulate annealing works and genetic algorithms don't. And in the end of this discussion, by the end of this discussion, sort of, we looked at each other, and we had this, which basically is what Nagylaki, I believe, mentioned before. What if sex is a mediocre optimizer of fitness? Okay, and let me sort of, I will stick to two loci, just because they provided me with a two-dimensional screen to project on. Okay, but everything I'll say sort of, also extends to multiple loci. So this is the fitness landscape of an imaginary two-gene organism, and the entries are the fitness of the combination of the genotype. And I'll tell you things that we know now, that A-sex will select the largest numbers. Okay.
So you're saying if you start with a population that's kind of covers the
all over, yes, right. If you start with a population that covers all over, yes. Everything else will vanish soon, and only these genotypes will happen. And eventually, sort of, 7 will also wither away. Okay, that's trivial. Okay, so it's physics. Okay. Now, it's proven. It's physics, and you can prove it. Okay. So now, so the interesting observation we made, and we called it mixability, is that sex does not favor these, but favors something else. Favors the alleles that perform well with many genetic partners. Okay, if you wish, it's going to favor the rows and columns with large averages. as opposed to the largest entries in this matrix. This is completely clear. Okay, so I'm sure that many of you knew that. Many didn't. So one of the reviewers of our paper said, "I will kick myself, for not having thought of this when I was a graduate student." Which is, in some sense, a very underhanded, a high underhanded high compliment. Okay, so this is something. So let me give you another sort of more rigorous. Imagine that your landscape is like that. Okay, that you have peaks, you have valleys, and you have a plateau. Okay. So you can actually prove, not hard, and that's again with Adi and Mark Feldman, that unless the peaks are more than twice the plateau will prevail under sex. Okay, so sexual evolution will miss these peaks because they're only by 90% higher than the plateau.
So this is the limit of high recombination, right? Is it or what?
It's the high recombination, yes, right. So everything else, the quantitative part of what I'm going to say is about high recombination, but similar things you can say about lower recombination. So sort of, these two will change for lower recombination. Okay, so this is one thing I want to set forth, all right? That there is, that we found a particular manifestation of what Nagylaki said before, namely that evolution does not necessarily optimize. Okay, so it does its own thing. Okay, so what I want to continue showing is that there is a computational way to get a little more better understanding of what evolution actually does. Okay, so so let me go to weak selection, which is sort of what is weak selection? It's again a point of view in evolution. It has to do with Motus Kimura's yes.
So are you done talking about the role of sex, or are you?
No. Will I ever?
No, I mean, because otherwise I had a comment,
but no, I want to hear your comment.
Oh, well, so the traditional explanation is that it's useful in coevolution, like for instance, against parasites.
True.
And
Yes.
From a game theoretic point of view, if you have an adversary who's not doing the optimal strategy, it's actually much harder to play.
Okay, so excellent, very good. So that's you said this is the traditional way of explanation. This is one of the 5 or 6 or 7 traditional ways of explanation. I mentioned the paper by Barton and Charlesworth. They sift through all these arguments and they find them inconclusive. Okay, so they find that none of them is clearly. So they look at them sympathetically. So they believe that one of these must work. Okay, so but this paper to me says that it's still a little open. So what? Okay. All right. So let me. You remember these landscapes that I showed you before? So these are like extreme rough landscapes. Okay, life is really hard here, right? So as Lars said yesterday, there is a very famous, 1969 stance in evolution by Kimura, saying that evolution is neutral. Okay, so then this has suffered friendly amendments. Okay, so since then, basically saying that it's not neutral, but it's sort of nearly neutral, and there is a mathematical incarnation of this near neutrality, which is called weak selection, And basically the weak selection says that the fitness landscape does not look like the sort of the extreme one that I've been showing you before, but it looks sort of like this, okay? Where the fitness of a particular genotype, and again, I'm talking about two, I'm showing you two loci, but what I'm going to tell you is true for multiple loci. The fitness of particular genotype is essentially about the same, all right? Well, plus a small number, which is much less smaller than 1. Actually, it's much smaller than R, the recombination ratio, okay? So, but certainly 1. And R min, okay, the minimum recombination ratio between any two loci that we are going to examine, okay, or two sets of loci, okay. But for two loci, it's 1 number.
But is it also much smaller than 1 over the size of the population?
It is about the size. It's not much smaller, but you think of it as smaller than the size of the population. Yes, it's about the same as well. You're right, yeah. But for what very much smaller than 1 is sort of is a good place to start. Okay. So there is a really important theorem of Nagylaki, 1993, who says the following: that under weak selection, linkage disequilibrium vanishes. Okay. Linkage disequilibrium for the computer science mathematicians in the audience is basically the distance of a distribution over a rank 1 distribution, of a product distribution. Okay, and the maximum, the minimum distance, and under weak selection, you don't have such distance. Okay, that basically these probabilities are essentially a product distribution if you forget a small error. Okay, and that holds after not log n, 2 log n generations. All right, so if where n is the number of alleles, total number of alleles.
Here also recombination is strong.
Right, yeah. So recombination. So everything sort of has things like 1 over r.
Yeah, so but like the higher
over r min.
Recombination, then like the inverse population size, then selection.
Right, yes. Sort of, yes, right. Yeah, but but that's the minimum recombination. The minimum recombination between any two sets, that we're talking about, sort of, and so on. Yes, so you're right. Yeah. So
The fact that the 1 over n there is crucial. It's not.
It's yes. First of all, this is infinite population, right? This is the infinite population approximation, anyway, right? But you are right. Okay, so you are right. 1 over s is yeah. Okay. The equations of the recurrence, it becomes like this. Okay, in other words, you don't want to track the genotype now, the genotype frequencies. You just want to track the allele frequencies. Is it clear? And the allele frequencies change like this. That the next day, the next generation, the allele frequency sort of is multiplied by this little thing here, which first of all, okay, it makes the point of the mixability thing that I told you, right? That I told you before, 5 minutes ago. Mixability says basically that evolution favors alleles that can do well with a wide range of partner alleles. And if you wish, this is what this says, right? It's that they increase by a factor which explicitly depends on how well they do with other alleles. Okay, so what I'm saying is that the Fisher-Wright equations, even for 2 genes, are complicated. Sort of, I enjoy staring at them, but sort of, but what I'm saying is that in linkage equilibrium they become extremely simple. Okay, so and allow you to, and this takes a little manipulation. All right, it's not completely direct. and allow you to make a few interesting remarks. One of them is what I just told you. Here's another one, okay, and tell me how you like it. So here's what this equation tells me, and what this equation should tell you, all right? That under weak selection, okay, the only assumption I've made, the evolution under natural selection becomes a game. Okay, I haven't defined what a game is, but how many would like me to? Okay.
Can I tell something before you say game?
Yeah.
Because are there non-trivial solutions, fixed point solutions of these equations?
Non-trivial.
Yeah.
Yes, actually. Yes, I mean,
Stable? Where the y's are neither 0 nor 1.
Yes.
Stable solutions.
Yes. There are. I mean, no, they are rare, but they exist. You are right.
There are deltas for which they exist.
There are deltas for which they exist. Yes. I mean, no, I'm in the middle of a debate about exactly this point. Yes. Right. Yeah. Okay. Yes, they exist. Okay. So, what is a game? By a game, I mean the following. It's basically what von Neumann, Nash, and so on call a game, which is it is a thought experiment whose purpose is to understand the rational behavior by rational agents. And a game is defined as follows. You have a finite set of players. Each player has available a finite set of actions. And if all players each chooses one action, this action combination imputes a particular utility on each one of the players. And this utility is known in advance. And basically what game theory does, it tries to understand how do these people behave, so what do you expect them to do, okay? So in the end you have equilibria, mixed strategies, Nobel Prizes, and so on, okay? Right, so, okay. All right. So here is something that I hope will surprise you, okay? I didn't know it a few months ago, and really, I hope you didn't know it. But under the weak selection, evolution is a game. Okay, who are the players? Important. The players are the loci. Okay, so if this reminds you of the selfish gene, think again. Okay, it's not that. All right, I mean, it's not at all that. So the players are the loci. The players are the loci. So every player has a set of actions, strategies. What are these actions? The actions, the strategies, are the alleles of the loci. Okay, so every player can choose alleles. As you know in game theory, players typically, occasionally, often, usually, use mixed strategies. Okay, in other words, they randomized over their inputs. Okay, so I have to answer now. The strategies are the alleles. What are the probabilities with which every player, that is every locus, plays a strategy that is an allele? And here's what it is. So by the way, I'm not making this up, okay? I am reading the equation I gave you before, okay? So all this is sort of, an imaginative reading, but precise reading of the equation I gave you before, okay? Putting it in a different framework. The common utility, in other words, here is what this makes it distinct from a selfish gene. That these loci, they have a common utility, okay? They strive for the same thing, all right? This is called a coordination game. In some sense, it is the simplest kind of game, all right? I mean, some people would call it no game at all, okay? But it is a game, okay? Game theory study, coordination game. And this game is only interesting when the players are cognitively challenged, okay? Because otherwise they could sort of, these are loci, right? I mean, they are cognitively challenged, okay? They cannot sort of, right. So the common utility is the organism's fitness, okay? That's what all the loci are striving for. To optimize expected organism fitness. All right? Let me repeat. I'm doing nothing else but read this equation. All this is from a game theoretic reading of this equation, nothing else. All right? All right? Surprise. What the players do, how the players behave, okay? They update their strategies. What the players do, they play it by the multiplicative weights update algorithm. This miraculous algorithm that I showed you before, okay? My friend Umesh, okay, with whom I wrote this, used to ask me, what algorithm would have created all this, in a mere 10 to the 12 steps, okay? Because that's, we heard yesterday, that's how many generations, I'll come to you. Yeah, that's how many generations we have had since the beginning. So, 10 to the 12 may sound like a big number, but it's not, okay? Since the beginning of my talk, your cell phone has done more than that. Has taken more steps than 10 to the 12.
But 10 to the 30 is a big number.
10 to the 30 is a huge
That's
number.
the other relevant number, 10 to the 30.
Yeah, 10 to the 30 is a great number, okay? If you think about it, 10 to the 30 is how much all cell phones in the world are doing in a year. They don't accomplish much, believe me.
No, but the 10 to the 30 is the number of parallel things.
No, yeah, so that's why I'm talking about parallel things. That's why I saw your bid and raised it, sort of, with parallel, with something parallel, okay? Okay. All right. So, okay. So, as I said, Umesh was asking me, what kind of algorithm could produce all this in 10 to the 12 steps? In some sense, we have an answer. Okay, we know what kind of algorithm. Not to read too much into this. So there is a lot that it leaves out. Okay, but sort of I find it extremely interesting that this algorithm that has created a shock of surprise to computer science over the past 10 years. Is you just find it when you look under the hood of evolution of natural selection. There's a little more.
There's a question up there.
Sorry.
Yeah, so I obviously haven't read your paper, and I can't necessarily do this in my head, but this thing that you just said, this is the levels of selection problem, and so the price equation formulation. It sounds.
No, it's not. It's not that. It's definitely not Price's equation. No.
Okay.
Yeah.
All right. It sounds like it.
Yeah. No, it's not Price's equation. It's not the fundamental theorem. Yeah, it's something else.
Okay.
Okay.
So you checked out like Stephen Frank.
I know. I know about Price equation. Sort of my co-authors know better than me. So that, yes, I know it's something else. Yeah. I will be delighted to hear from you if you see a connection. Okay. Any questions? So as Nagylaki said, don't tell me it's the fundamental theorem. Okay. Okay. So there is a little more, which I think is sort of remember, I wanted to tell you a few things, a few random thoughts that I've had about the role of sex, and sort of frankly, this equation, this algorithm, is enabled by sex. Without sex, you don't have this. Sex is at the basis of the long chain, sort of that creates this algorithm, recombination. Okay. So here is something that a couple of computer scientists knew. It was written obscurely in a few papers, in a couple of papers. But sort of, we, if you want, sort of, I can give you a very rigorous derivation. Okay. The same equation, in other words, the multiplicative weights update applied to allele frequencies, can be reinterpreted as an online optimization problem. Okay, so here's what I mean. Let's make the following thought experiment. All right. You say, I bet that each locus at each generation is trying to optimize something. Okay. So let's reverse engineer. What is it that every locus is trying to maximize? Is it clear? Are you together? So and there is a very crisp mathematical answer to this, and it entails variation in evolution. Okay. So here is what it does. At each generation, each locus is striving to maximize as if the sum of two things: the cumulative expected fitness of the organism over all previous generations. Okay, so it's sort of trying to optimize something that looks like sort of looking too much to the back. Okay, and it's a little puzzling to us. Okay, so the first thing means that what happened 100 generations ago is equally important to the locus as what happened the last generation. Okay? So it's a sum, okay? No weights, nothing, okay? It's a sum. It's a straight sum. The cumulative expected fitness of the organism over all previous generations, plus 1 over S, which is a huge, of course, it's a huge multiplier, times the entropy of the alleles distribution. Okay, so what I'm saying is that implicit in the equations of life under weak selection, in the equations of evolution, of natural selection, under weak selection, is the fact that what these thingings are trying to optimize, to maximize, one of them, given actually a lot of weight, and the smaller the selection strength, the bigger the weight, is the entropy of the alleles distribution, okay, which to computer scientists is a synonym of variation. Okay? Yes?
That's reminiscent of the free energy, which is the sum of enthalpy and entropy, where enthalpy here would correspond to the fitness.
Could be sort of I'm sure that this has a physics analog. What I'm saying, what I'm saying is this, okay? I'm saying the following precise thing: that if you write this plus this, okay, and you take the KKT conditions and you try to maximize it overall PIJs, okay, overall XIs, okay, overall sort of allele frequencies, overall set of allele frequencies. So basically what you are doing is you are maximizing overall possible vectors of allele distributions at each generation. Okay, you are optimizing over this. The following, it's the sum overall T, overall up to now, okay, sorry, of essentially the expected fitness of the organism, which is xi times the fitness of at time t of expected fitness of allele I at time t, plus 1 over s, if it is minus 1 over s, the sum overall I of xi ln xi. So this is, I rewrote this, okay? Now what I'm saying is that this is a simple concave maximization problem. It's a complex problem. strictly convex problem. It has a unique optimum. You can find it, but it's a convex because it's something linear times minus something convex.
So it is exactly the free energy.
Sorry?
It's exactly the free energy.
It's exactly
The free energy.
It's exactly the free energy, you are saying.
The
Okay.
expectation of I of the energy minus the entropy.
Expectation of.
The energy minus the entropy.
So you
To maximize the energy.
you have omitted something, okay? That I'm adding over all previous generations.
No, but so the problems you talked about earlier, one can also think of the time as being a direction. If I think of it as energy of a polymer, I mean, then it becomes a similar thing, but it's just the time in the.
So
time is
listen.
not time in the free
Listen,
energy.
okay, so you know
It's
the-
length.
Yeah, I'm not. You know, I'm not saying that this is new. Okay, so I'm saying that computer scientists have discovered it. Okay, about a few years back. What I'm saying is that so I want to see if it's exactly what you are saying. So we do have physicists in our as co-authors, right? But
I'm sorry.
sorry.
I'm sorry to hear that. No, but wait, the fitness term is summed over time, but not the trajectories.
No.
You're not maximizing the entropy of the trajectories, the final point.
I'm not exactly. So I'm maximizing right now. So I'm talking about right now.
The fitness takes the whole time, right? But you're not maximizing the entropy of the full trajectory. It's just the endpoints.
Okay,
So I'm not.
I see. No, that's.
Okay. So I mean, if it's identical with something, I'll be equally delighted. So I'm telling you, it's
Certainly not obvious identical.
Yeah.
Okay, no,
So it's identical with something that Manfred Wartmuth has said but didn't say it very clearly in a paper about 10 years ago. It's also identical to something that Lorenzo Recchia has said in another paper recently. Okay, but I don't have pride of discovering this. I'm just I'm just very interested in it. Okay, I'm keenly interested in it. Right? Because it sort of tells you that variation sort of is in some sense an obsession of the beast. Okay, so it's part of what it tries to optimize. Okay. Any more questions? Okay, so this is sort of. You want to see these derivations, or it's just differentiating this stuff and putting some Lagrange multiplier.
Yes, can you please, please?
Sorry.
Can you?
Okay, sure, yeah. So basically, what you want, you also have this, okay, and xi negative, but even ignoring this, or ignoring the Lagrange multiplier for this, you basically get that what the KKT conditions would give you is that Fi, I believe, equals 1 plus ln xi. And basically, this will give you that. Sorry? Sorry. T is not is an index, right? Yeah, thanks. And basically, this will tell you. So, am I doing something wrong here in the differentiation? So, solving this will tell you that xi, because that's what we are talking about. Xi t plus 1 is sort of, to get the normalization, you have to look at the previous t, but it's essentially xi times 1 xit plus t sorry, to the x.
I'm sure the tau is T.
Sorry?
X is T, then tau.
X is, no, tau is the running index. To get this solution, you have to rewrite this for t equals t minus 1
Okay.
and subtract. X of e times, s times s times fi tau, which is this. Sorry, not 1 plus x, which is again 1 plus s times fi tau, which is exactly this. And I may have missed a sign somewhere. But, and also I jumped over a derivation where I rewrite this for t equals t minus 1 and subtract. So, again, looking at this and comparing with this, what it tells you is that the Fisher-Wright equations in this particular case, they optimize this convex combination, which again has a prominent term entropy. Okay. So, I mean, doing this brings back memory. I mean, I've done this before. I've done this sort of like years ago. So this is not, you know, I'm sure that this is a very common derivation. But what I'm saying is it also applies here. All right. So let's change the subject. All right. So I don't know, how many of you know about pointer dogs? So, you know, they are, okay. All right. So pointer dogs are dogs trained so that when there is life in the environment, they point it, okay? So a pointer dog in this room will be totally confused. Right? This particular pointer dog is pointing at young Charles Darwin. Darwin was very interested in pointer dogs, so interested that he had befriended the shady characters in the London suburbs that raised them. And the way they raise pointer dogs is sort of interesting. They take good hounds and they train them to point. They select the best pointers. They breed them. They teach the puppies how to point. And after a while, they have created better dogs that point. Okay? Very interesting. Okay, so this particular pointer dog is pointing at Cyril Waddington. Okay, one of the greatest experimentalists of the 19th century. Right? So, and what I want to tell you is an experiment that Waddington did. So he took so you can tell a computer scientist because he calls these animals flies. Okay, you know, so I'm a computer scientist, right? So he took a bunch of flies, and that was in Edinburgh in I believe in the 1980s, 1950s. And what he did is he raised the temperature from room temperature to 20 degrees, to 40 degrees. Actually, he cooked the puppet for a while, to 40 degrees. And as a result, something changed. Okay, so you know what really changed was not the head did not become red as I depict, but their wings became less cross-hatched than they should be. Okay, so there was some kind of phenotypical change, some kind of notable phenotypical change. So about 15% of them changed. So what he did is he repeated this, and then he bred only the changed flies, and suddenly 60% had changed. Okay, and then he continued. He bred again, and something like 63% changed. So the changed flies kept increasing until they became all of the flies had changed. And then he did something else. He lowered the temperature, and in the next generation, about 25 of them had changed, which to me is very surprising. Okay, this is a phenomenon that was called, I'm not sure that Waddington called it, but it's called genetic assimilation. Adaptations to the environment become genetic, okay, that become heritable. And I mean, the more you think about it, if you're not sort of, a true biologist, sort of, the more you see the sort of, the specter of Lamarck. Right, so that this is something really fishy going on, okay? And of course, there is also the Siberian foxes, that we have heard recently and so on, right? I mean, so that's, but don't worry, okay? So there is a question: is there a genetic explanation? Can you find a purely genetic explanation? This is sort of, the mental exercise I want to take us through, and there is, okay? So so what you want to do is you want to find a function, let's say a Boolean function, of two variables, of several variables, a genetic variable and an environmental variable. H means high temperature. It's also 0, 1. Okay, so so basically you want X is many variables, many Boolean variables, many 0, 1 variables. You want the probability, in the beginning you have, let's say that all xi's are 0, 1 with probability half. In the beginning, the probability, the sort of, the starting probability is 0%, okay? So, you will never see a changed fly because the temperature is low. Then you hit the puppet, and suddenly you see that 50% of by the same probability the flies are changed. Then you breed them, you breed the changed flies, and you see that the next day 60% have changed. And in fact, it's significant that this is slightly bigger than 50%. Okay, if you do the calculation after, you see that it's sort of magically like 57, 60%. After successive breedings, becomes 100%. But then if you lower the temperature from 1 to 0, it stays to 25%. Okay, so this is what you want to achieve, all right? I mean, so this is the specifications of the function I want to create, right? Of the Boolean function I want to create in these, let's say, 11 Boolean variables, 10 genes and an environmental variable. Okay? And it's a very simple function. Anybody wants to guess? It's a threshold function, right? So here is the incidentally, I should have told you this. In computer science, you love Boolean functions, all right? And we a lot of my work, even though it's the first time I'm mentioning here, a lot of our work is about Boolean functions. Okay, so we had to indulge when we saw this, all right? I mean, so in any event, suppose that redhead is this Boolean function of 10 genes and high temperature. That redhead means that the sum of the 10 genes plus 3 times this variable is 10. Okay? Suppose that this is our function, all right? This is what redhead means. It turns out, suppose that the genes are independent random variables, a little hard to swallow, but not in terms of after the weak selection slide I showed you before, with pi initially half. So If you buy all this, all properties of the Waddington experiment are satisfied. Okay, so basically you create exactly the Waddington's curve. Okay, with these parameters. Is this clear? And so I was really very happy, very proud when I figured this out. And then Adil Ivnat pointed out that in 1958, a naturalist called Stern, in the General American Naturalist, had essentially proposed exactly the same. Yeah.
Is 10 the smallest number you need to fix the
No.
or you just-
I don't know. I haven't looked much into that. Yeah, good questions. I don't know. So I'm curious. Yeah, I should be curious. What's the best fit? Okay. So that's. So this is. This is, I think, a nice exercise. And the exercise that you could predict that computer scientists who have taken. Who have gone through. But then. Here's what got into us, okay? We said, how about arbitrary Boolean functions, right? How do you call it? This is something that Adil Ivnat had always been interested in. Okay, can we say something about very sort of arbitrary functions of genes, all right? Besides the sort of simple threshold functions, right? So, what if we have an arbitrary Boolean function of genes? And in fact, now we dispose of the environmental variable H. Okay, there is no environment, right? There is no environmental input, right? We only let the population evolve, right? Without sort of having a control, an environmental control over it. And suppose that the satisfying genotypes have a fitness advantage. In other words, the genotypes that satisfy are, let's say, slightly above, are slightly more fit than the non-satisfying genotypes. Okay, in Waddington experiments, of course, the satisfying genotypes had an absolute advantage. Okay, because they survived. Right, everybody else was wiped out. Okay, will this trait be fixed eventually? Do you expect this trait to be fixed? Of course, if this is a non-satisfying truth assignment, okay? This is a unsatisfiable formula, a satisfiable Boolean variable function. This is not going to happen, okay? So you must have at least a way of satisfying it, all right? In fact, you should have some reasonable probability of satisfying it, okay? In other words, if you have a large population, a few people should, a few members of this population should satisfy at random this formula. In any event, suppose you have such a Boolean formula, but otherwise arbitrary. Will this trait be fixed eventually? Okay, and so, of course, this is computer scientists starting from a nice biology experiment and taking a tangent. But it turns out. So this is another story I want to tell you that. In our group, we proved a very interesting theorem about Boolean functions. Right? That the answer is yes. You can do this for every Boolean function. All right. So this is work with Adil Ivnat, Aviad Rubinstein, a student, Greg Valiant, former student now at Stanford, and Andrew Wong, who is a fellow this semester. And the theorem says that any Boolean function of genes which confers a non plus epsilon selection advantage will be fixed. And when I say will be fixed, I mean with high probability within polynomial mean generations and with polynomial population.
Polynomial, the number of genes?
Polynomial number of genes.
Epsilon is some function of the number of genes in the population?
What's epsilon? The probability of satisfaction.
Yeah.
The probability of satisfaction, let's say, yes, so it's polynomial in the number of genes and 1 over epsilon. Okay?
Right, so if there's just one genotype,
Right, then you are cooked. Yes,
Yeah.
right. Yeah. Or if there is no genotype, then even more. Okay.
and I'm not allowed to even cluster all the satisfying.
No. So
they're uniformly distributed.
No.
around the
They're not any function.
The satisfying ones, I can.
Could be clustered. but they don't have to be.
What's a polynomial population?
Polynomial is like n squared or n cubed. In our case, it's n to the fourth. So it's basically it's.
Finite population result?
So it turns out it's a finite population result, but the population does not have to be incredibly large, okay, for this to work, okay. It will work in the limit for incredibly large populations, but it only needs to be n to the fourth, where n is the number of genes, and maybe also multiplied by 1 over epsilon to the fourth, where epsilon is like 1/10 the probability that a random genotype has the trait.
But you're saying you can fix epsilon and then take n large.
Right,
This is right.
right. So it's polynomial. So it's, frankly, if memory serves, it's 1 over n. It's n over epsilon, or if you wish, we call it pi 0, the probability that in the beginning you satisfy, raised to the fourth power. And I may be slightly wrong about the 4. Okay, I don't remember if it multiplies both or not.
That's how long the time is.
That's and how large the population has to be for this to take hold.
Can I just ask an intuitive question? Let's suppose whatever n you choose, I choose pi 0 to be 1 over n.
Right. Fine.
Then it seems to me...
N to the 8th.
Well, anyway, no, but we know in that case that it won't necessarily go to fixation. Right?
Well, yes, because you are going to miss it in the beginning.
Yeah.
You are going to miss it.
No,
in
I'm
the
just trying to fit that
No, sorry.
important evolutionary scenario with this one.
Yeah, I understand. So here, I think I misspoke. I didn't explain something. N is the number of genes.
Yes, we're mixing up the n here in the
Oh.
finite population.
Yes, right.
Quite a lot.
Yeah. So N is the number of genes. How do you want? What variable you want to use?
So there's no population.
So this is the population.
And the time is also
Also, yes.
And then the
Yeah.
pi naught is the probability that satisfies.
Pi naught is probably the random genotype, or the fraction of genotypes that satisfy.
The fraction of genotypes, of Boolean genotypes that satisfy. So if it's small, then of course you have to have a large population to sample, okay? Okay, and I'm not sure that this is accurate, and I'm sure it's not optimal, okay, that it can be brought down. So, okay.
It just seems strange because, like, I could say, I don't have pi. I want to say, like, I add a bunch of totally irrelevant genes.
I want my theorems to sound strange, by the way. Okay?
That's good. Yeah.
I don't want to say, I can say, of course, yeah. I love that. Okay, so tell me.
No, I was taking pi naught to 0 in my head.
Yeah.
Okay, never mind.
Okay.
Well, it sounds strange to me because it seems to be inconsistent with Haldane's result that advantageous alleles are fixed with low probability.
Are fixed with.
Low probability.
So I'm not. So this does not say that alleles will be fixed. There are many ways to fix this. This is a multigenic trait that has many ways of many ways of getting there.
So it's the phenotype that's fixed.
So it's the phenotype. The phenotype will be fixed.
This is infinite combination.
Sorry, it's a combination. Yes, infinite combination. Yes.
At least one. The phenotype will reach this.
The phenotype will reach us. Yes, right.
Yes, I guess the thing which comes in here is what are in toy situations, what are reasonable assumptions about how pi naught depends upon the number of genes.
Yeah.
And that's,
So
and that's one of these ones about how many solutions there are and so on.
right.
Which is a very
So for threshold functions, it's sort of usually it's huge, right? I mean, it's like a big part of the cube. Yes, right. But for other functions, maybe smaller. But we want it to be like reasonably large for this to work.
Do you know what the
final population looks like? So is there a lot of diversity in the final population, or is it?
No, there's not. You know, so in our proof technique, you lose also genetic diversity. But I suspect that there is a different proof technique that gives you a much better result, and also maintains genetic diversity. In other words, you know, others, you know. Yeah, good point. Okay, so I can't believe you guys let me finish my talk. Okay, you know, so quickly, you know, this, you know, like what it was experiment. I mean, this does not involve mutation, but basically it involves, as you know, a strange manipulation of allelic frequencies so that a phenotypic property emerges. All right. So I started explaining what computer science brings to the table, and you know, I think it's a lot. I think I'm really proud of what you bring to the table. It does not compare what you guys bring to the table. Okay, so, you know, you bring to the table the world's most fascinating, most important problem, and a lot of brilliant work done on it for the past 100 years. But sort of, you know, you can, you know, I hope I convince some of you that computational insights can be reasonably productive. Sort of, you know, they can at least make you think in a slightly new way about things that you knew. So I'll tell you two random problems that jumped to my mind, so, you know, this morning, because I thought I should put some of the problems. Is there an analytical proof of the mixability principle, of the fact that that alleles that are mixable prevail, are favored? And is the fact that I told you, sort of, you know, the implicit entropy maximization, a more general phenomenon in evolution, which I guess, as I gathered from our discussion 15 minutes ago, many of you believe that it is. All right, so
Any questions, more general computational speculations?
The question of coming back to the Leventhal paradox, which has nothing to do with evolution, but what you're talking about here does. But if I say, well, okay, the natural guess is that for interesting phenotypes, the pi naught is exponentially small in various things.
Mm-hmm.
And so that this
You
really...
think so? You think that you
Well,
think?
I don't know. I have no idea. If one sort of thinks that somehow evolution is solved pretty hard problems, that you have to beat out things that are already there, but you can do that by making a new function and winning on that instead of winning something else. So it seems like one of those sort of questions in you know, sort of toy models that go you know, beyond the simplest ones, because single Boolean function, and in ones where one may not be able to prove things, but maybe we'll start getting some insights and go forward on that. Because this is really, you know, getting to the issue of the very large number of possible solutions.
Right.
Where it's coming from.
Of course, yeah.
Which, at least my sense is, I find it hard to find things about that in the literature, evolution literature.
With a large number of solutions.
Yeah, and emphasizing.
I see.
You know, presumably relevant for why genomes are the size they are, or why
Right.
they don't need to be any bigger. I know that or at a smaller scale, why, you know, 20 amino acids is enough.
Right.
Proteins besides they are. So there's a lot of sort of things in there which one maybe can use some of that information.
Okay.
Also, the fact that recombination rates are very low. Also somehow relates to this issue. You know, what is it that sets the recombination rates?
Mendel didn't stumble on that, but yeah.
Well, the low recombination rates, I mean, you're kind of talking about that here, right? You're saying
Right.
you don't want to, huh?
This is high recombination.
I've
no,
been
but-
talking about higher combination rates.
Yeah, but you said, like, you know, it's not so good if you, these higher combination rates are not so good actually for finding the good solutions for increasing fitness.
I guess right, yes, but I mean, no, but right, okay. So I mean, no, I talked a lot about genetic algorithms, right? I mean, no, and that's not what I'm interested in, okay? I mean, no, so and actually, genetic algorithms are using the genetic metaphor in order to solve optimization problems. If you look at the literature, you'll see that the researchers are more mesmerized by evolution than interested in solving actual problems. Okay, so that this is what this is what, and I am like that. Okay, so right, yes, if I really need to solve a problem, I would not use recombination.
But maybe you wouldn't use very high recombination necessarily.
You are saying that
and
maybe,
so maybe this is connected to why recombination rates are not infinite in actual populations.
Right. Okay. Could be.
So the recombination rate is a relatively easy thing for organisms to change around. Like, these factors
Sure.
of 10 and long time scales much larger. So it's microbes by enormous factors, and it's one where the sort of balance of that of the advantages of recombination and the advantages of not having so much. One
Yeah,
very
so-
interesting quantitative question.
Right.
Yeah, I don't know how easy it is for viruses to manipulate.
Depends on the virus.
Yeah.
So part of what Christos is saying, though he's too humble to say it himself, is that he's advocating a bit of a shift from the notion of evolution as being optimization, survival of the fittest, to being maybe something different. So survival of mixable traits, survival of modular traits. That if you accept sex as a fact of evolution, then what you get is not something which says that the organisms around us are the fittest, but rather the organisms around us are the ones whose genomes are the most mixable.
But this is something where it really depends on how much sex we're talking about.
Right.
Like whether sex overwhelms selection or whether selection will shift the composition of what genomes are out there, and then what is mixable changes, right?
Sure, but the picture changes once you take into account that maybe fitness is not the goal of evolution.
I don't know about what the goal, but I'll just say like the strong recombination picture is.
I think the problem is fitness is not one thing. It's some extremely high dimensional object, and certainly the other way, which is maybe related to this thing at all, is that the big advantage of sex is with time dependent environments, and therefore the fitness is time dependent, and many different problems that it may solve, and it's the keeping the diversity of the population is the role of sex, which you talked about, but then also back aside again.
Well, I wanted to talk about a couple of things. But yes, yeah, right.
Or even without time dependence. Just on the way to the goal, when you actually need to produce the best solution, it helps to have more variation.
Right. Yeah. Yes, so in the very end, at equilibrium, very likely, actually, most likely, there will be no variation at all. Is that?
Yeah, I'm from an outsider's perspective, I just have a naive sort of question, which is maybe sort of from where I can see, you've got a bunch of different people who think about different things accumulated for a semester, and what are kind of major, in your sort of ideal world, what would come out of, what are the kinds of questions that would have some sort of answers to them by the end of the semester?
So the ones that were discussed and then they had put in the beginning are amazing. Are amazing questions. That's the role of sex, how is variance maintained, how novelty emerges, maybe what is modularity and how it emerges. So these are great deep questions. I'm sure there are 4 of 20, and what I want to be achieved, solving, answering, definitely a few of them would be amazing, okay, creating an environment where people forge lifelong ties, research ties, people from who wouldn't do so otherwise, would be also an excellent outcome.
I'm going to try and ask a similar question tomorrow. Force people to do it.
One could add, so for example, Monty discussed in the first talk the problem of variation, right? How to explain the amount of variation that exists in nature. And that's a very big problem even today, and evolution has not yet been resolved. And then the same problem came up again at the end of Rasmus's talk, right? When that neutralist selectionist debate came up yet again, even though it's been going on for a very long time, and there is yet no resolution for it. The question came up of how much, for example, of the Drosophila genome and how much of the human genome is experiencing natural selection, and whether that and what explained the extent of genetic variation that exists. And as an example, Christos just presented here one new way of thinking about the maintenance of genetic variation that has not been proposed before. So it really exemplifies the kind of contributions that one can make to the study of evolution. Does that make sense?
Covers me. Yes. Yes.
I think in some sense, your goal in one way is too immodest, in that one's not going to solve these questions. But it seems to me but it's too modest in another way. It's something what I would like to come out of this is a much better framing of questions. So these questions which all these people frame, some mathematical, very limited way, some in these big picture ways, and turning some of those, or preferably even more new ones, into questions which one can think of a well-defined answer. And then, of course, figure out which of those might be interesting from the biology. But it seems that to really advance the field, one needs a lot more, which you've talked about some of doing,
right,
of trying to really frame the questions well. And I think that's, there's a much too much tendency, and this is the bit I didn't like about your talk, of going and starting with all the dead white males and quoting from them. Right? And they're the very big picture questions, but what's needed, the very big picture and the very small picture in the sense of the population genetics, the mathematical questions of the underlying framework of that. It really is the sort of all the things in between, and really framing those questions in a way to try to move forwards.
Agreed.
That's a way to break it.
Maybe just a little bit on Marty's point. So in terms of this role of diversity, so certainly one can see that diversity plays a role when there's uncertainty about the future. To try to try to make sure one is hedging against possible futures. But I think one of the things that came across in this, one of the features of what Christos was talking about is that it comes up also for a computational reason, which is that you're exploring an environment computationally, and it's difficult, and so this notion of diversity or regularization, that you don't want to fit the part of the data that you don't want to take the part of the search space that you've already explored and take it too seriously, and therefore you want to maintain as much diversity as possible to do this, just as a computational mechanism for optimization. This is an additional feature that comes across.
Right.
I mean, this kind of reminds me of the survival of the fittest versus survival of the flattest tension, which is that balance. So do you specialize for your current circumstance, or do you remain a generalist,
right,
genomically? And
sure,
it seems like that depends on mutation rate and recombination rate.
Yeah, I talked about echoes of this. Yes, right.
Yeah.
Very clear.
So I'm wondering if you accept the results of the statements of your theorems as a ground truth, how much of the what was known?
How much we have checked the proofs?
No,
we have checked the proofs.
No. I'm sure you did. Everybody does, but at the end. So my question is, how much of the what was known in biology you can actually get? I mean, if you accept as truth the statement of the theorem, how much of what these other fields have investigated? If you have thought about this.
Okay.
How much of what was known as in evolution or in genomics you can recover?
Listen, okay, sort of, I've been in this business for a long time, okay, there are so many ways to answer what you are asking, okay, so so the question is, there is something hubristic, arrogant, about proving theorems about anything, right? Because the question is, that theorems are something that you cannot dispute, and yet, they have a an unreasonable power. So in formulating your theorems, you should be extra careful. And of course, what can I say? Here is one little thing that I want to say, right? In what a theoretician does, is prove theorems about what other people know empirically, and in the best of all worlds, proves a theorem that utterly surprises them and moves them all in a different direction, moves the school of fish in a completely different direction. What happens usually proves something that people say, yeah, I knew all that. Thanks for making me sure. And the vast majority of theorems I have proved. So in my career, out of the latter kind.
Is that what you were asking?
Sorry?
Not exactly. So I'm asking basically, there are some, let's say let me use the term axiomatic principles. If you accept this as ground truth, what you have the statement of the theory, can you actually recover what was originally known? People were suspecting, oh, we have this model, can you actually recover? Can you actually go the other direction? Not starting from something and proving it, but accepting what you have stated as a theory. If you can obtain the axioms that they were, the models that they were actually having.
So I think you. So we have to take this offline. So maybe you give scientific fields too much credit. They don't have axioms, right?
No, but if you are to develop a computational view of biology, and you can say evolutionary biology, then you can say we can actually redo, starting from this, we can actually redo everything you were doing. This gives more legitimacy to the field.
Yeah. So that's not, that's not my style. That's not what I intended to do. That's what I set out to do. That's what I said in the beginning of my talk. Okay. I don't want to sort of. I don't want to say that I have sort of the ultimate truth and let me rewrite your books. Okay. So that's not what I admire the problems that they admire. And I try to do. I try to investigate them in my style and using the things that I know.
Just to make it clear, I'm not implying anything. I'm just stating what I want to say. So it's not.
You are yes. But I think your question sort of builds some kind of picture that I don't recognize.
I didn't imply that. I'm just curious if there can be a foundation. A more unified foundational study. That's what.
Okay, so I don't know. You are young, okay? So in the 1940s, okay? So people started sort of engineering, okay? Sort of mechanical and civil engineering, right? They said let's build an axiomatic foundation of a complete axiomatic foundation of civil engineering, okay, so they're called the rational mechanics, okay. It was a huge field, okay. A lot of very smart people, very smart mathematicians, applied mathematicians worked on this. Okay? It did not have a lasting impact, okay. So it's not, these are lofty projects, okay. So but.
Christos had a list of fields at the beginning of his talk that have been profoundly changed by contamination by computer science ideas, but primarily the way they've changed has not been via some axiomatization that's come from computer science. And you might have to look at each of the individual fields to see quite what aspect of computing they've benefited from, but I can't think of a single example where it's been axioms.
There's also, the general question of, the role of being able to solve some models or being able to prove things about some models is one, it sort of somehow is the basis of generalizing from that. And there's certainly the experience of condensed matter physics, which is when people really started looking at simple models and trying to understand those, rather than saying one was going to learn things from Schrödinger's equation, was when the field started really to take off and progress. And then ideas of the sort of understanding of why, when there's lots of big numbers around, certain things can be universal or pretty generic, and the sort of hope of those, and the getting, some intuition from the things that one can solve exactly, or sometimes prove things about, and so on.
Right.
So it seems, that, because what you're, the models and even some of the things you're doing, except the fact that you're proving them, has a lot of overlap with, with approaches that some physicists might, might take, and which models come naturally is different, and somehow which is, I think, one of the big benefits here is that the kind of models one might start with are very different, and then one has a similar question about them, and that back and forth.
Incidentally, I did very little to introduce the next speaker. So you are going to see a lot more of the computer science mindset, in the next in Varun's lecture.
Okay, so let's maybe break for lunch now, but so come back this afternoon for a discussion of the origin of complexity and how complexity can possibly evolve, computational account of that. Okay, so let's thank Christos again.